Members, welcome to TEP Education. We are still looking at biology, and now we are focusing more on classes of enzymes. Classes of enzymes. So, we have already seen that enzymes are biocatalysts. Enzymes are biocatalysts. If they are biocatalysts and protein in what? In nature. Now we want to see how do we classify these proteins? How do we classify the enzymes? Category 1, we can say enzymes can be classified according to the following. According to the following. What are those categories? Category A, according to the area of operation. What do we mean here? Where an enzyme catalyzes the reaction from, if it catalyzes a reaction within a cell, so you're going to call it intracellular enzymes. So you can say these are enzymes. These are enzymes that catalyze reactions within the cell that do produce them. So they work within the cell that produces them. So we can continue and say, get some examples of this. Examples of such enzymes. The first one we shall take are 1, digestive enzymes in holozones. To understand what holozones are, those organs that have a digestive system. So digestive enzymes in holozones, all of them, rather take another, that would be an extracellular enzyme. We want to take the one of intracellular enzymes. We shall talk about glycolytic enzymes. Glycolytic enzymes, the ones that control glycolysis in the cytoplasm of the cells. We take a very many of them. We would even take glucohexokinase. Hexokinase enzyme that catalyzes the phosphorylation of glucose. That would be 1. 2, we can even take electron transport system enzymes. Also, we can also take Krebs cycle enzymes. These are vital chemical reactions that take place within our cells to gain energy from organic food substances, majorly glucose. The other category, Roman number 2, would be now extracellular enzymes. Extracellular enzymes. These work outside cells, outside the cells that produce them. We can now go and say they are produced by glands in secretions. So the ones we had earlier put here, the digestive enzymes. Digestive enzymes in the gut of holozones. Earlier said that holozones are organisms that have an internal digestive system. So what are those enzymes we can take? For example, salivary amylase. Salivary amylase that acts on starch. It is from the salivary gland. Our salivary glands produce saliva. One of them, the parotid gland, produces the salivary amylase, which is the content of the saliva. That's why we are saying they are produced in secretions. In most cases, secretions do not contain a single ingredient, but rather more than one ingredient. We can talk about pepsin, produced by the gastric gland. Gastric walls, or you can call them glands. A gland is a group of cells or a cell that can make secretions, majorly either enzymes or hormones. We can even talk about pancreatic lipase from the pancreas. You know, the pancreas is a double gland, exocrine gland at the same time endocrine gland. The exocrine part of the pancreas produces pancreatic lipase together with the pancreatic amylase. Pancreatic amylase together with the trypsin. These are enzymes from the pancreas. So now we can see that category. Let's continue further and see which other category can we put here according to. Let's continue with that. Enzymes can be classified according to the area of operation. We have seen it too. Intracellular enzymes and extracellular enzymes. Then, which other category can we use to classify enzymes? To classify enzymes further, we can continue and say, according to the chemical reaction they catalyze. According to the chemical reaction they catalyze. Which chemical reaction does an enzyme catalyze? Because enzymes do catalyze, and we said catalysis is altering of the rate of a chemical reaction. The first ones we shall take are oxidative oxidoreductases. You see our suffix as is coming back. So oxidoreductases, these catalyze the oxidation or reduction of a substrate. We have very many oxidoreductases in our bodies because most of the chemical reactions that take place in energy synthesis are oxidation reactions and reduction reactions. An oxidation reaction is this. If I have BH and I continue, remember they catalyze reversible reactions, and I remove BH from this and then form the hydrogen ion also there. If I remove the hydrogen ion, remove of a hydrogen ion from a substrate is what we are calling as reduction. So this would be a reductase enzyme. Or we can go further and call it a reductase. That is a very big group. We call it now a dehydrogenase enzyme. So it has removed the hydrogen from a substrate. That's why collectively we can call them oxidoreductases. Let's even add an S there, because there is no single oxidoreductase, but rather a group of them. If it did the opposite, and we find when B plus has received the proton and now we are receiving BH, it would mean that is an oxidase enzyme, but a special one we shall call hydrogenase enzymes. Hydrogenase enzymes. So meaning collectively, all enzymes that work in such reactions that involve removal or addition of hydrogen on a food substrate or in a substrate, we shall collectively call them oxidoreductases. We can continue to number 2. We have transferases. It is a group. Enzymes concerned with the transfer of atoms or group of atoms, transfer of atoms or group of atoms from one substrate to another, from one to another. We have special of them, the ones that do transamination. The one which is done in the creation of non-essential amino acids. Now those ones we shall just call them transaminases. That one is done in the liver. During creation rates of isoleucine from the essential amino acids, you will find there is transfer of an amino group from one molecule to another. So we shall collectively call those ones transaminases. But there can be more when it is just lifting of a phosphate group, an organic phosphate group, from one molecule to another. We shall call those ones still transferases, a group of enzymes concerned with the transfer of atoms. 3. We can have hydrolases. Hydrolases, these break down. These break down bigger molecules to smaller molecules in presence of water. In the presence stroke addition of water. So such enzymes, when you are talking about them, like now the digestive enzyme, let's say if you are talking about salivary amylase, we don't usually say catalyzes the breakdown. We could say catalyzes the hydrolysis of starch to maltose. The other word comes in. Digestive enzymes are hydrolases. They catalyze the hydrolysis. They catalyze the breakdown of big molecules to smaller molecules. Starch is far bigger than maltose. That's why we're calling them hydrolases. We can take one more and let's say ligases. These catalyze the union of smaller molecules to big molecules by use of energy, by use of ATP. If we combine glucose plus glucose to create maltose, we shall say that one is a ligase enzyme. So there are very many ligase enzymes in plants and even in animals. By the way, we already said enzymes work within living things. They can work outside living